Hey everyone, today's notes are just going through passive transport. All right, so the idea between passive transport has to do with molecules moving from high to low concentration, right? So first things first, we can talk about selective permeability just as a vocabulary sense. Selectively permeable just means that the membranes will select what passes through. Typically speaking, your membranes will allow small nonpolar hydrophobic molecules like oxygen and carbon dioxide to pass through the membrane freely, going from high to low concentration. Okay. There are some molecules that cannot get through or are not selectively or cannot pass through the membrane. Those are going to be your hydrophilic molecules, polar molecules, larger molecules, charged particles or ions. Examples would be sugars like your glucose, as well as water. Okay. So in terms of transport, there's two main types, passive and active. Again, today's notes are just going to be talking about the passive types. So passive transport is the movement of molecules from high to low concentration, or moving down the concentration gradient or down the electrochemical gradient. And this does not require energy. They do them on their own. Okay. That will be in terms of bringing materials into the cell and out of the cell. And examples of passive transport are going to be your diffusion, your osmosis, and your facilitated diffusion. First one's diffusion. Diffusion is, again, from high to low. It moves down the concentration gradient without the use of energy. This can take place in gases, liquids, and solids. Gases are going to be your perfumes or colognes or air fresheners. Your liquids are going to be your movement of like dissolving a sugar cube throughout the liquid. Okay. And then your solids will be, say, sitting in a chair and the heat from your body diffuses into the chair, so your chair is warm when you get up. All right, so this is just molecules showing how different molecules move at a rate. Remember, they're going to move from high to low concentration. Okay. There's just different rates of diffusion depending on what types of molecules they are. Now we're going to get into osmosis. Osmosis is just a diffusion of water from high to low concentration through a selectively permeable membrane. Okay, so it's essentially the diffusion of water is another way to say it. All right. You'll notice in this picture here, water, okay, it shows it going from left to right, okay. It shows sugar molecules. Sugar molecules cannot pass through that selectively permeable membrane, only water can. Therefore, water wants to go to high to low concentration. If you note right here on the left side that water is at a high concentration because there's more water per sugar solute. On the left side, there's less. So water's higher concentrated on the left. Therefore, it wants to move to the right to equal out your concentration of water and sugar. Lastly, we have facilitated diffusion. Facilitated diffusion is going to be molecules moving from high to low concentration through a membrane with the assistance or the facilitated by proteins, transport proteins. These are going to be going through those cell membranes. This is going to allow molecules like your ions, your polar molecules, your larger molecules, right? Like your sugar to get through. And there's going to be two types. There's channel proteins and carrier proteins. We're about to talk about those. And each one is specific for each substance. We call them subject specific, meaning for every molecule you have, it has to have its own specific protein channel. So I'm going to pass this slide. Okay. Channel proteins, okay, are going to provide more of an opening for molecules to go through. A good example is aquaporins. Typically, they're only open to water, but typically they also are open all the time, so water can just pass through from high to low concentration. Some of which are gated. Gated channels are going to be more like they're only open when there's a stimulus that allows them. This is going to be seen more often in muscle contraction. Then there's carrier proteins. Carrier proteins, the difference are they're not always open. They're going to undergo conformational changes. Okay, an example of this is going to be glucose transporters or protein transporters through the membranes. And that's it.